Hi everyone, this is Swetha from Telangana, India, pursuing final year BTech in Agriculture Engineering from College of Agriculture Engineering, Madagascar. First of all, I am very thankful to Acharya Engineering Agricultural University and College of Agriculture Engineering, Madagascar for selecting me for undergraduate inbound credit mobility program at University of Malaysia, Kelantan under the integration of University of Malaysia, Kelantan and Acharya Engineering Agricultural University under the institutional development plan and my parents for supporting me to get this great wonderful opportunity. And presenting you is my the PowerPoint presentation in which I have done internship for 1 month. So these are some personal details includes which includes name, matric number, education, university, and my mentor is Mohammed Shaiful Azman and my field supervisor is Associate Professor Dr. Fatima Khayat and the topic assigned for me is plant tissue culture and the period of training is 1 month. And this is about the arrival of us at UMK City Campus, and we have arrived at UMK City Campus on 14th February, and there was a welcoming ceremony held at UMK City Campus Kotabaru, and on February 15th we have reached to UMK Delhi Campus where we have worked on plant tissue culture topic for about 1 month, and these are some photographs related to the arrival session. And this is about the location of our training site. This is the plant tissue culture lab which is located in UMK University of Malaysia Kelantan Delhi campus, and the plant tissue culture lab mainly consists of 4 rooms, which is cleaning room, media preparation room, aseptic transfer room, and growth room, and an office. And this is the nursery which is located beside of the plant tissue culture lab, in which variety of orchids are grown such as dendrobium, oncidium, cymbidium, spathoglotis, vanda, etc. And about plant tissue culture is a collection of techniques used to maintain or grow plant cells, tissues, or organs under sterile conditions on a nutrient culture medium of known composition. And there are many stages which are included in plant tissue culture, such as selection, initiation, multiplication, rooting, and also acclimatization. So, the figure shows the process stages of plant tissue culture. And about the orchids are plants that belong to the family Orchidaceae, a diverse and widespread group of flowering plants with blooms that are often colorful and fragrant. The first flower shows the Spathoglotus plicata orchid, and the second figure shows about the distribution range of Orchidaceae family. And my topic mainly is about Spathoglotus plicata orchid, which I have done training for about 1 month. And about Spectoglotis, it is commonly known as purple orchid and is a genus of about 50 species of orchids in a family Orchidaceae. They are evergreen terrestrial herbs with crowded pseudobulbs, a small number of leaves, and medium-sized resupinate flowers on an upright flower stem. The petals are all similar to each other and are white, yellow, pink, or purple. So these are some scientific classification of Spathoglotis plicata bloom, and this is for the micropropagation of Spathoglotis plicata. In vitro clonal propagation of plants from shoot tips or nodal explants, usually with an accelerated proliferation of shoots during subculture, is known as micropropagation. And about the culture media in plant tissue culture provide water, mineral, nutrients, needs, vitamin, growth regulators, access to atmosphere of gas exchange and medium for removal of plant metabolite waste. The culture media contains inorganic such as macronutrients and also organic compounds such as sugar and also vitamins, plant growth regulators and agar. Murashigan's cook medium is most suitable and commonly used basic tissue culture medium for plant regeneration. There are three different media are prepared for practicing subculture of Spathoglotis explants. They are multiply media, elongation media, and rooting media. All these three media are prepared by the same process but except the use of the type of the PGR and the concentration varies from 1 to 1. And this is the process of media preparation. First of all, we'll take a flask of 2 liter and we'll place it on a magnetic stirrer and we'll add 600 ml of distilled water. Then we dissolve the 30 grams of sugar into it and by next 50 ml of macronutrients and followed by 5 ml of micronutrients and followed by 5 ml iron stock and 1 ml of vitamin stock. And in the case of multiply media, we add 1 mg per liter of BAP, and in case of elongation media, there is no PGR is added, and in case of rooting media, 0.5 mg per liter IIA is added. Then the pH was adjusted to 5.8. Then the volume was adjusted to 1 liter bearing the distilled water. And then the 8 grams of agar was added and melted in the solution. And then the solution is placed into the micro oven for 6 minutes. So after the 6 minutes, the flask is removed from the micro oven and it is placed on the magnetic stirrer for 2 to 3 minutes. After the 2 to 3 minutes, it is filled. The medium is filled in the jars and it is autoclaved. And these are some photographs related to the media preparation of Spathoglotis plicata bloom. So about the subculture of Spathoglotis explants, then subculture is a process by which the tissue or explants is first subdivided and then transferred into fresh culture medium. Preparation of plant tissue for tissue culture is performed under aseptic conditions under HEPA filtered air provided by a laminar flow chamber. Therefore, the tissue is grown in sterile containers such as petri dishes or flasks in a growth room with controlled temperature and light intensity. So these are some photographs related to the subculture of the Spathoglotis plicata bloom. So about acclimatization. Acclimatization here simply means the adaptation of plants to a new environment. Most species grown in vitro require an acclimatization process in order to ensure that sufficient number of plants can survive and grow vigorously after being transferred to ex vivo soil. So in case of any contamination in the in case of any contamination in the orchids like by fungus or bacteria, then the following steps are to be followed to remove the bacteria. So these are some photographs related to the acclimatization of the contaminated plants. And these are the other activities which I have performed and done in the 1 month training session, such as ginger sowing, nursery housekeeping, and these are some photographs related to that, like preparation of media for filling pots, nursery cleaning, ginger sowing, and rearrangement of pots, weeding, nursery visit, and at the last conclusion. And it was a great experience for me to learn about the new things which I have not known previously about the plant tissue culture. It is totally a new topic to me, and I have learned many things by hand-on experience, and also about the media operations involved in the plant tissue culture, such as media preparations, subculturing, and acclimatization, and also hardening, and about the nursery management. So it was great opportunity for me to learn about the new things and also do the hand-on experience. So thank you.